Arachnological and Biogeography of the Gondwanan family with the corresponding name that I cannot pronounce. So it's all yours, Sam.
Can you see me?
Yes, we can.
Okay. So, hello, my name is Shahan Derkar-Abadian. Despite the title of my talk, I will not have time to discuss systematics of this group. This is all going to be about biogeography. So, I'm lucky enough to be one of the people on this planet who study the family Triaenonychidae. This is a family of Apiliones that is relatively diverse, about 105 genera, and somewhere around 460 species and subspecies are currently described. This family is entirely found in the Southern Hemisphere after our revision. So they're pretty much found on all the former landmasses with former Gondwana. So this is a pretty interesting distribution. So the point of this talk, or the point of this project, is to really kind of go after the biogeographic history. So the first thing we have to do is build a phylogeny of Triaenonychidae. And if you know me, you know I go UCE. So I use ultra-conserved elements to kind of build my phylogenies. And ultra-conserved elements have allowed me to include museum specimens. So in this phylogeny, I have some museum specimens that are over 100 years old. So that's pretty good. As far as taxon sample, we currently have about 75% of the genera sequenced, which is pretty phenomenal. We're missing about 27 or so. Most of these missing genera are monotypic genera that are described or diagnosed based on really highly variable characters. So we're not too worried about missing them. And if we needed to, or actually we could place those missing genera, most of them, into this phylogeny purely based on morphology. So we have a fairly complete taxon sample. The only taxon we're missing that is pretty important to this question excuse me, is this genus, or this species called Permectostethys unifalculatus. This is an endemic to the Crozet Islands of the Southern Indian Ocean. This is obviously a long distance dispersal event. We were not able to get specimens of this species, but we're working on it. But for now, we have excellent taxon sampling. So getting into biogeography, there are 3 points I want to make. First is dealing with the organism. Triaenonychids, like many Apiliones, are dispersal limited, and they have high microhabitat specificity. This kind of specificity and limited dispersibility makes Apiliones really good biogeographic indicators and candidates for studies like this. Second point I want to make is about the region. In taxa that inhabited Gondwana, 200 million years ago, we can expect to see biogeographic history that's explained by vicariance because of the continental breakup, and sometimes in younger taxa, long distance dispersal is involved. In taxa with biological characteristics like this, we expect to see vicariance due to continental breakup. Finally, the last point I want to make has to do with analyses. So I approach this in using a model-based approach in a likelihood framework so that we can kind of tease apart some of these different scenarios. So this is my interpretation of Gondwanan breakup, continental breakup. This is essentially a timeline of events. So up top, we have 200 million years ago. At the bottom is present time. So a lot happened. What I do want to point out is the beginning of continental breakup is typically dated to 180 to 170 million years ago. So right about here. So there have been a lot of studies focusing on Gondwanan taxa, and I'm going to point out a couple of them right now that have used arachnids, because of course. So the first is in spiders, orsilobids. They're found on 4 major land masses that are essentially part of former Gondwana. And if we look at the phylogeny and their distributions, we essentially see that every regional fauna is monophyletic, which means this is a relatively easy story to tell. It's kind of simple, it's clean, it's nice and friendly. Next, we have in Apiliones, in the family Patalidae, again, former Gondwanan distribution. Again, what we see with their current distribution patterns is that this is a relatively easy story to tell. Not all land masses are monophyletic, but it's essentially pretty simple. So what does it look like in the Triaenonychidae? Not easy, it's quite complicated. And of course, I get stuck with this one, but I'm happy to work on a complicated system like this because that's what research is about for me. So what we see here is essentially not a single landmass is monophyletic, with the exception of New Caledonia, and that is because there's only 2 species known from New Caledonia. Even though I just said that, we have a study from our lab group that is soon to be published with a denser taxon sampling in this part of the tree that does not recover a monophyletic New Caledonia. So it's complicated. And, you know, my task is now to explain this very complex scenario in hopefully simple terms. And I've kind of had to draw evidence from multiple places. So obviously phylogeny and taxonomy, divergent states, plate tectonics, distribution of these taxa, paleoclimate, current climate, vegetation, all of these things to kind of form a cohesive picture to explain this complex scenario. So I'm going to attempt to do that and tell part of this story today. And I'm going to start by doing that, or I'm going to start doing that by explaining or giving the conclusions first, so that this can be in your head while I go through the results. And I think there are multiple factors that can explain the distribution of Triaenonychidae across Gondwana. And these are not mutually exclusive, of course. First, we have divergences that predate the breakup of Gondwana. Second, paleoclimatic gradients and mountain building I'm considering as well. And on the biotic side of things, in taxa that are low dispersal, high microhabitat preference, we expect to see a general pattern of common vicariance and rare dispersal. Finally, kind of underlying all of this is the idea of niche conservatism. These taxa have a preferred niche, a preferred microhabitat, and they have a tendency to stay there through geologic time. So first, I'm going to talk about divergence dating. So here's time-calibrated phylogeny of just Triaenonychidae. This red bar indicates the onset of continental breakup of Gondwana. What we can see in these two earliest diverging nodes, even when including 95% confidence intervals, they are estimated to occur before continental breakup. So I'm going to switch this phylogeny to biogeographic reconstruction. And here the nodes are reconstructed ancestral ranges. And I'm going to focus in on these 2 earliest diverging nodes and their descendants. So each node has a likelihood pi. And each piece of this pie tells us the likelihood for a particular ancestral range. So at this earliest diverging node, we have 50% likelihood that the ancestor had a range of South America and South Africa and East Australia. There are other pieces of this pie that suggest East Australia and either South Africa or South America. And so we have our Gondwanan map. We have a high likelihood that this ancestor had a distribution that was something like this. I'm not sure how far south or how far north. The important thing is the east-west. So when we look at the descendants, we see here one is estimated as South America plus South Africa, and the other is East Australia. So we end up with a distribution that's kind of like this. And the question is, why was there vicariance in this lineage when continents were all together in its contiguous paleo vegetation? And the answer, I think, lies in a combination of paleoclimatic gradients. So first, paleoclimatic gradients. We see in the north, there's this kind of winter wet, and then some warm temperate. Winter wet is still habitable for Triaenonychids. And in the south here, including East Australia, is this little pocket of cool temperate. So I think that's part of it. The second half, I think, has to do with mountain building, specifically the transantarctic mountains. So if we look at East Antarctica, there are a lot of mountains there. And I think this, in combination with paleoclimates, help to kind of get divergence initiated prior to breakup of Gondwanan. So I'm going to take this idea a bit further and go a little deeper in the phylogeny and focus on these two clades. So one in green and then this other one in orange. The ancestor to these lineage, or these clades, is estimated to be in South America and South Africa. If we go a little deeper into each, we see transitions from South Africa to Madagascar to Australia. And here we see South America to Australia and eventually to New Zealand. So at some point in time here, we can reconstruct their distributions of these two clades as like this: South Africa, Madagascar to Australia, South America to New Zealand to Australia. So now you can kind of see how things are coming together. And if we kind of take a step back and think about mountains again, we can see how mountains fit into this picture very well to help separate and keep these lineages diverged. And then if we bring back in paleoclimates, I would make a guess that this lineage is perhaps a warmer temperate lineage, and this is a cooler temperate lineage. Again, climate helping to kind of keep these things separate. So getting onto the idea of common vicariants, this particular phylogeny, I only show nodes that are reconstructed to be vicariant events. You can see there's a lot of them. This red box indicates the kind of major time period of continental breakup for Gondwana, and you can see there are a lot of vicariant nodes associated with this, which is what we expect. What about rare dispersal? So there's one instance of long distance dispersal in this phylogeny, and that's to New Caledonia. New Caledonia separated from Australia 80 million years ago. After that separation, it went through multiple periods where it was completely submerged underwater, re-emerging 37 million years ago to essentially where it is now. In the original analysis I showed you, that node, so New Caledonia is nested within New Zealand. That node is estimated to be a vicariant event, which is impossible because from a biogeographic perspective, New Caledonia is an oceanic island. So why? Well, the problem is because these original models don't include jump dispersal. The good thing about BiogeoBears, which is the program I'm using, you can include this +J parameter, which is jump dispersal. And so when we include that, the likelihood of the analysis increases and this node switches to a dispersal event. So here is the rare dispersal. Finally, I brought up niche conservatism. This is still a work in progress. I'm trying to figure out exactly how I want to approach this question, but the idea is, based on current distribution of these taxa, I would like to code them according to their climate classification now, and then reconstruct ancestral climates in the past to see if those ancestral climates match what we expect based on reconstructed ranges and predicted paleoclimates. So in the case of these two clades, I would hypothesize that this clade would be reconstructed as warmer temperate, and this would be cooler temperate. So that's essentially what I have for now. This, again, is still work in progress, so we're still trying to figure this out, but that's what I have for now, and thank you for your time and joining me in my apartment, and I'll hopefully take questions.
Thanks, Shahan, for a great presentation, fascinating group. Shahan has indicated he might not hear me very well, so I'm going to post the questions as I talk about them. So from Brian Patrick, what do you find if you used the typical genes we've all been using for 20 years, the usual suspects, compared to the UCEs? Would you expect to find differences?
So with the usual suspects, so that previous paper I mentioned with kind of showing non-monophyletic New Caledonia was based on the usual suspects. And we kind of see the same thing. A lot of the backbone nodes are not well supported, but we kind of see the same thing. There are no land masses that are monophyletic. Although I would say UCEs are, well, no, it kind of depends on the gene, but UCEs are pretty conservative in some cases. So we get the same picture. We just don't have high enough resolution to kind of deal with that, or high enough support to deal with the questions at the kind of very base of the phylogeny.
Okay, you're getting a lot of positive comments. How was the dating done?
Oh, how was it dated? So I had a lot of outgroups I didn't show, and it's all dated based on fossils. There was four fossils I used that range in age from 405 million years for the root, some at 305, one at 99 million years, and then another at 48. So it was pretty good spread as far as times for the fossils. I actually did a second set of analyses that included like a 2 million year calibration that was based on biogeography to kind of prevent divergence dates from going too far back, and I essentially got the same thing, at least for the deeper nodes. So.
Okay, thank you, Shahan. We are basically at the end of our session. Thanks to all the